Hello everyone! Today we are going to explore an essential instrument used in biology, histology, and cytology laboratories, the light microscope. Let us begin by thinking about a simple question. Many biological structures, such as cells, bacteria, and microscopic tissue components, are too small to be seen with the naked eye. So how do scientists observe them? The answer lies in the use of microscopes. A microscope is an instrument designed to produce magnified visual or photographic images of objects that are too small to be seen by the unaided human eye. In simple terms, the microscope enlarges tiny objects so that we can study their structure and details clearly. To achieve this, a microscope performs three important tasks. It magnifies the image of the specimen, it separates the fine details of the image, and finally, it makes these details visible either to the human eye or to a camera. In this video, we will explain how a light microscope works, show its parts, and demonstrate magnification, resolution, and visibility with real slides. First, magnification. Magnification enlarges the apparent size of the specimen so small features become visible. We describe magnification as the product of the objective and eyepiece powers, for example. 40 times objective times 10 times eyepiece equals 400 times. Next, resolution. Resolution is the ability to distinguish 2 points as separate and determines how much detail you can see. Resolution depends on wavelength of light and the numerical aperture of the objective. Shorter wavelengths and higher numerical apertures improve resolution. Finally, visibility. Contrast techniques like staining, phase contrast, and dark field increase visibility of structures that would otherwise be hard to detect. Together, magnification, resolution, and visibility determine whether a structure can be observed clearly and accurately. Now, we look at the major parts of a compound light microscope, starting with the eyepiece, where you view the image. The objective lenses sit close to the specimen and provide the primary magnification. Microscopes include multiple objectives with different powers. The stage holds the slide, and the condenser and aperture diaphragm shape and focus the light that illuminates the sample. The coarse and fine focus knobs move the stage or objective to bring the specimen into sharp focus. The light source, often an LED, provides stable illumination. Adjusting light intensity can improve contrast and clarity. Optical path recap. Light passes from the source through the condenser, through the sample, into the objective, then through the eyepiece, to your eye or camera. Preparing a slide starts with placing a thin sample on a glass slide and covering it with a cover slip to protect and flatten it. Staining uses dyes like methylene blue or gram stain to add contrast and reveal specific cell structures or bacterial groups. When you begin viewing, start with the lowest power objective to locate the specimen, center it, and then increase magnification. Use coarse focus at low power, then switch to fine focus and higher power for sharp detail. Always refocus after changing objectives. For very small bacteria or internal cell structures, use oil immersion objectives and proper technique to achieve maximum resolution. Throughout, maintain clean slides, correct illumination, and steady sample placement to avoid artifacts or loss of detail. Let's see examples. Bright field images of stained cells show nuclei and large organelles, while bacteria require higher magnification and staining. Phase contrast and dark field techniques reveal unstained live cells and motile bacteria without dyes by enhancing contrast. Remember limitations. Light microscopes cannot resolve structures smaller than about 200 nanometers, so electron microscopy is needed for finer detail. Calibration and scale bars ensure measurements are accurate. Always note objective power and camera settings when capturing images. In summary, the light microscope magnifies, resolves, and makes visible tiny biological structures, enabling scientific observation and discovery. Thank you for watching. Practice with real microscopes, explore prepared slides, and keep observing. There's always more to discover. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, Connecticut, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.